All right, welcome back. Today we're tackling something absolutely fundamental, maybe the most fundamental mission for all of life: how to make more life. We're going to look at two wildly different strategies. One is like making a perfect blueprint, and the other, well, it's more like rolling the dice in a giant genetic lottery.
So, before we get into all the nitty-gritty details, let's just zoom out for a second. Think about it. Every single living thing, from the tiniest microbe to a massive blue whale, what's the one core purpose that connects all of them?
You got it. It's reproduction. That's the biological process that lets life create, well, more life. It's how the story continues, how that incredible legacy gets passed from one generation to the next. Without it, life on this planet just stops. Full stop. Okay, so to get this all-important mission done, life came up with two totally different game plans. We can think of them as the blueprint, that's all about making a perfect, identical copy, versus the lottery, which is all about shuffling the genetic deck and seeing what you get. One needs just a single parent, the other, well, it takes two.
All right, so let's jump right into that first strategy. This is the direct, super efficient world of asexual reproduction. We're calling it the asexual blueprint for a reason.
And the idea here, it's really simple. You have one parent, and it produces offspring that are genetically identical to itself. Yeah, it's basically like a biological photocopier. There's no mixing, no shuffling, just a perfect, flawless clone every single time.
Now, if you look at the microscopic world, the most common way this happens is through something called binary fission. And honestly, it's pretty much what it sounds like. You have one cell, it gets bigger, and then it just splits into two identical halves. And boom! Each one of those halves is now its own new, complete organism. This is the absolute go-to method for little guys like the amoeba or the paramecium. You gotta understand, for them, cell division is reproduction. It's one and the same. It's ridiculously fast, it's efficient, and it just gets the job done. No muss, no fuss.
Okay, check this out. Another really cool method is called budding. With this one, the new individual doesn't come from the parent splitting in half. Instead, it literally grows right off the parent's body, kind of like a little sprout or a bud.
You see this with single-celled stuff like yeast, where these tiny buds just form and pop off. But it's not just for microbes. It also happens in multicellular creatures like the hydra. A little mini-me version of the parent just grows right on it, gets big enough, and then breaks away to start its own life.
And you know who the real masters of this asexual game are? Plants. Oh yeah. They use something called vegetative propagation, which is just a fancy way of saying they create new plants from their own parts. That potato in your pantry, that's not a seed, it's a tuber. The onion, a bulb. These are living pieces of the parent plant, just waiting to sprout into a brand new, identical clone.
Okay, let's completely shift gears now. We're leaving behind that clean, predictable world of perfect copies. It's time to step into the complex, kind of messy, but absolutely amazing world of sexual reproduction. The sexual shuffle. And the key difference here, it is profound. Instead of one parent making a perfect clone, now you have two parents, and each one contributes genetic information. And the result? Offspring that are totally, completely genetically unique. They're different from their parents, they're different from their siblings. This right here, this is the engine that drives diversity. So yeah, this whole process is way more complicated than just splitting in two. You can actually think of it like a play in 3 acts. First, you have the preparation, then the main event, and finally, the development that comes after. Let's walk through each stage.
So, Act 1, pre-fertilization. Before any of that genetic shuffling can happen, these special reproductive cells, we call them gametes, they have to be created first. And then, well, they've got to somehow find each other.
And these gametes, they come in a couple of different flavors. In some simple organisms, like algae, the gametes actually look pretty much identical. We call those homogametes. But in most animals, including us humans, they are very different. You've got the male gamete, the sperm, which is super tiny and designed to be a great swimmer, and then you have the female gamete, the egg, which is huge in comparison, stationary, and packed full of nutrients.
All right, on to Act 2, the main event. This is that crucial moment, the fusion of these gametes. It's called fertilization, or syngamy, if you want to get technical. This is it. This is when two halves of a genetic story come together to create a completely new, totally unique individual.
But how does this fusion actually happen? Well, life's come up with two main ways to do it. First, there's external fertilization.
You see this a lot with fish and amphibians. They just release their gametes out into the water and hope for the best. It's a numbers game, and it's super risky. The eggs are totally exposed. Then you have internal fertilization. This is what most land animals do. It all happens safely inside the female's body, which gives the developing embryo way more protection.
So, once fertilization happens, you have that first single cell, which is called a zygote. But the story is far from over. In fact, this is where Act 3 begins, the incredible journey where that one single cell divides and transforms into a whole complex organism.
And that journey has a name: embryogenesis. It's not just about cells dividing over and over again, though there's a ton of that. It's also about something called cell differentiation. This is the magic part, where cells start getting special jobs. One becomes a heart cell, another becomes a nerve cell, another one becomes a skin cell. All of them working together, building an entire embryo from that one starting zygote.
Now, where this development takes place separates animals into two big groups. First, you've got oviparous animals. Think birds, reptiles. They lay eggs. So the embryo develops outside the mom's body, but it's protected by a shell. Then you have viviparous animals. That's us and other mammals. We keep the embryo inside, giving it protection and nutrients the whole time, right up until it's ready to be born.
It's not just animals either. In flowering plants, you see a really similar transformation after fertilization. The zygote becomes the embryo, the part around it, the ovule, hardens into the seed, and the ovary itself, that swells up and becomes the fruit. It's this brilliant trick to get animals to eat it and then, well, spread the seeds around.
So, as we start to pull this all together, we come right back to that fundamental trade-off we talked about at the start. On one hand, you have the asexual blueprint. It's fast, it's simple, it's efficient. Perfect copies every time. On the other hand, you have the sexual shuffle. It's slow, it's complex, it takes a lot of energy. But the payoff? Oh, the payoff is huge. Endless, incredible genetic diversity.
And that leaves us with one last big, fascinating question, right? If the blueprint method, cloning, is so much faster and easier, why is our world so full of creatures that use the slow, complicated sexual shuffle? Why did evolution favor that strategy for so many species, including us? Well, the answer is all about the power of diversity. Think about it. In a world that's always changing, where there are new diseases or new predators, a population of identical clones is incredibly vulnerable. One weakness can wipe them all out. But a diverse population, one created by that genetic shuffle, it's got a much better chance. Someone in that group might just have the winning lottery ticket, the right genes to survive and carry on. It's not about being efficient in the short term, it's life's ultimate long-term insurance policy.